I'm glad to have the opportunity to talk to you about the development and regulation of medical devices. This session complements two other talks in the series, one on preclinical device development and one on the regulation of drugs and devices. I think most folks have an idea of how drugs are regulated, but for many, the regulation of medical devices simply seems like a black box. And part of the reason is because the medical device regulation encompasses such a wide range of products, everything from things as simple as IV tubing up to catheter-placed heart valves, software, complex imaging machinery, and then also in vitro diagnostics, just the lab tests that are done. So there are differences between drugs and devices that sort of illustrate the reason for these different approaches to regulation. First of all, drugs are pure molecules. If you change one atom, you've got a different drug. Devices, on the other hand, are complex components, and it's possible to change some of the components or alter them while still having essentially the same device. So you might modify a product in response to some issue that arises, but it's still basically the same device. In drugs, we're concerned about toxicology, whereas devices, the issue is biocompatibility. Drugs have a short half-life and a very long market life, whereas devices are durable equipment. It may be an implant that's going to be around for several decades, and they have very rapid product cycles. With drugs, we're concerned about drug interactions and administration of the wrong drug or the wrong dose. In devices, the issue is more a malfunction of the device or user error in its operation. Drugs typically require large clinical studies, whereas devices may be approved on the basis for marketing approval, may be approved just on the basis of bench or animal studies. If clinical studies are required, they typically are small to moderate size, much smaller than what's required for the usual drug application. So during this talk, I'm going to talk about the device life cycle and developmental process. You may have seen some of that already. I'll touch on devices, the definition of them, drugs, and combination products. I'll review safety and effectiveness and their definitions. And then just for completeness, so you understand the environment of the device world, I'll review briefly device classification and the different pathways to market, just to give you a complete picture. I'll then touch on off-label use and development strategies. Again, we'll have heard more about that on the preclinical presentation. And then I'll get into the investigational device exemption regulations and interacting with the FDA. And my purpose here is not to make you an expert in FDA regulations, but rather to inform you on how CDRH thinks about things, how it's organized, and importantly, how to recognize when you're doing a device study and how to interact with the FDA so you can have your clinical trial move forward expeditiously. So the device product life cycle begins with the concept, goes to prototypes, preclinical studies, potentially clinical studies, marketing applications, manufacturing, entry into commercial use, and then all too quickly with devices, obsolescence. Now you notice that there are lines connecting all of these because as products go from one iteration to another, everything you learn on this first cycle influences how products in the next iteration of a device are developed, how they're regulated, and how their behavior is monitored once they're on the market. And so there's a pipeline. There's constant churn in medical device technology over and over and over. And everything we learn with each cycle impacts what happens in the regulation of devices and review and development in the subsequent cycles. So there's a science and a regulatory cycle. So the science cycle is one of design, engineering issues, biocompatibility, material sciences, understanding the mode of action. If there's a clinical trial, then clinical sciences related to design and conduct of a clinical study. There are environmental studies, quality systems, factors related to marketing, and then commercial use, epidemiology, adverse events reporting, post-market surveillance, risk analysis, and again, the end of the product life. There's a regulatory cycle that follows along with this. There's a request for designation. There may be the question, is this even a device? And we'll talk about that. There's an FDA site called Device Advice that helps you through all of this. We'll talk about that. There may be early planning meetings with the FDA where you discuss the preclinical studies, the clinical studies, and your marketing studies if you're a manufacturer. And there are ways to get input from the FDA to help you through this. Then there are the investigational device exemptions, agreements with the FDA. There are a number of guidance documents. I'll touch on those. There are marketing applications. We'll review those. And then I'm not going to talk about post-market issues, but in the post-market realm, there may be post-market studies. There may be issues that come up that lead to warning letters, safety alerts, recalls, or, as I said before, modification of a device to address any issues that may have arisen. So we'll turn now to devices, drugs, and combination products. So this is the definition of a device from the regulations, and I'm sorry, but I'm going to read most of it. So a device means an instrument, apparatus, implement, machine, and yes, even contrivances, implants, in vitro reagents, or other similar articles, including any component, part, or accessory which is intended for use in the diagnosis of disease or other conditions, or in the cure, mitigation, treatment, or prevention of disease in man or other animals, or is intended to affect the structure or any function of the body of man or other animals, and which does not achieve its primary intended purpose through chemical action, and which is not dependent on being metabolized. So just to summarize that, it's basically anything remotely medical that's not a drug or device. And I'm belaboring the point here just to emphasize that when the medical device amendments were enacted that began regulation of medical devices, the intent was to capture everything, not a narrow slice of devices, but to make sure that all medical devices were captured in terms of being subject to the device regulations. So the devices classifications are listed in FDA database. So if you're developing a product or a device, you can look in the FDA database and find out if it's already classified. If it's a new device that's not been classified, then this is a little repetitive, but it's an unclassified device. But you can look in the product classification database, and all these things I'm showing you, they're easily found online. If you search FDA product classification, this will pop up. So here I've searched for magnetic resonance. When you do the search, it turns up with magnetic resonance coils and imaging systems. And this tells you the device classification for marketing applications is class 2, and we'll come to that. And it tells you the regulation number that defines magnetic resonance devices. So if we go to this 892.1000, this is the regulatory definition of a magnetic resonance diagnostic device. So you do need to keep in mind that when we use the word device, it refers not just to the physical device or a virtual device, if it's software, for example, but it refers to the device and its indication for use. From the FDA's perspective, these are inseparable. They are inextricably linked. So if you consider biliary stents, for example, biliary stents are indicated for the treatment of biliary strictures or narrowing of the bile ducts. These are typically placed in patients with cancer whose bile ducts are obstructed. They have a very short lifespan remaining potentially. And in the environment in which those stents are placed, it's one, there's not a lot of movement and there's no exposure to blood. There's no issue of clotting. It's a pretty forgiving environment. On the other hand, if you use the same device, a biliary stent, but you implant it in a peripheral artery to treat narrowing of an artery, then it's a new device. It's the same physical item, but it's a new indication for use. And I think you can appreciate that this device designed for being placed in a bile duct, when you put it in a dynamic arterial environment where it's exposed to blood products and the clotting cascade and potentially movement, if it's and flexing repeatedly, if it's across a joint, there's whole new risks and benefits that need to be considered. So what about combination products? You know, science marches on, and now we have drug-eluting stents and drug-eluting discs. So on the left is a stent that's intended to be implanted in an artery, and it's coated with a drug that once it's implanted, the drug comes off and it prevents the the artery from going in to the stent struts and closing the artery off again. The drug-eluting disc is a disc which contains a chemotherapy drug, and it's implanted in a brain tumor, and then the drug is released over time. So are these drugs or are these devices? So the primary mode of action for the stent is that the stent opens the artery, and the drug is a secondary action where the drug is presenting inflammation and restenosis. The drug-eluting disc, the primary mode of action there is cancer chemotherapy for a brain tumor, and the secondary action is for local drug delivery. And so the primary review center, whether it's the Center for Drugs or the Center for Devices, depends on the primary mode of action. So the drug-eluting stent would be regulated as a device, and there would be input from the Center for Drugs. The drug-eluting disc, on the other hand, would be regulated as a drug because it's primary cancer chemotherapy, but there would be input from the Center for Devices because of the device component, which is the drug delivery disc. So now we'll talk about safety and effectiveness. So the Food, Drug, and Cosmetic Act gives FDA explicit authority to ensure that devices are both safe and effective before being marketed. This is in contrast to the period before the medical device amendments were enacted, when the agency could only respond to hazardous devices after marketing had begun. So safety means that the probable benefits to health for its intended use, when accompanied by adequate directions on how to use it and warnings against unsafe use, outweigh any probable benefits. So risks. So the benefits, given adequate directions, have to outweigh any probable risks. There has to be an unreasonable risk of illness or injury. So this may require in vitro studies or in vivo studies, and that may be sufficient depending on the device, but there may also be a requirement for clinical investigations to demonstrate safety. Effectiveness, on the other hand, means that in a significant portion of the target population, the use of the device for its intended uses and conditions of use will provide clinically significant results. The point here is that you can't make something that carries out an action and show that it does that action, but if there's no benefit, there's no effectiveness, then it really is not suitable for marketing. So it has to have a clinical benefit. Otherwise, there's nothing to outweigh any risks. And this is typically shown through well-controlled investigations. And I would mention here that well-controlled investigations does not necessarily mean a randomized controlled trial. It could be a historical control trial. There may be performance criteria that it can meet in a single-arm study potentially. So now I'll just briefly review the device classification for marketing purposes and the pathways to market. I said I'm not going to use a lot of jargon, but there are two things that you'll hear. One is 510K, which is one pathway to market, and the other is PMA, which is a second pathway to market. And we'll touch on those because you will hear those if you're in the device device environment. So medical devices, as I indicated earlier, vary widely in complexity and potential risk. So devices are classified based on the basis of the nature of the device and the extent of FDA control to ensure safety and effectiveness. And these are class 1, 2, and 3 in order of increasing risk. Certainly the IV tubing that I showed earlier requires a totally different level of oversight compared to a transcatheter heart valve. So the class 1 devices are at the lowest level, and these are simply subject to what are called general controls. So general controls, and there are others beyond what I've listed, but the top ones are that it prohibits adulterated or misbranded devices. It requires adherence to good manufacturing practices and requires registration by manufacturers. So things as examples are lead shields that you might find in radiology to protect people from radiation. Some operating room tables are class 1. And almost all class 1 devices are exempted from submission of a marketing application prior to marketing. The next step up is class 2. So these are subject to the same general controls as for class 1, but in addition, there are special controls that are established by regulation because the agency needs more oversight of this class of product. So these controls may be published performance standards. They may be a requirement for patient registries. There may be a requirement for post-market surveillance. There are often guidance documents that outline specific non-clinical and potentially clinical studies that need to be done. So the main approach to a Class 2 marketing application is that you want to show substantial equivalence to a predicate device. So the predicate device is something that's already on the market, and the goal of a successful 510K submission, 510K is the section of the regulation that governs this. The successful 510K submission will simply has to show that your device, the new device, is as safe and as effective as whatever's already on the market. It's in a sense a Me Too device, although there's some more latitude related to that. And a 510K submission is required for most, but not all Class 2 devices. So the imaging systems, for example, are Class 2 devices. So CT scanners, even the latest greatest CT scanner, ultrasound machine, MR system, those are all Class 2 devices. And these IVC filters, which are implanted in the vein that drains the lower half of the body to capture clots and prevent pulmonary embolism, those are Class 2 devices also. And finally, there are Class 3. So these are subject to the same general controls as Class 1 and 2, but here, the idea is that the general and special controls really cannot provide reasonable assurance of safety and effectiveness to permit marketing. Typically, these are going to be implants, heart valves, for example, or devices that support or sustain human life, like a ventricular assist device, which is an implant that takes over the work of the heart pumping the blood. And these are ones that potentially present a potentially unreasonable risk of illness or injury. So for these products to get to market, these require an approved pre-market approval, or PMA application, before they can go into commercial distribution. So these are required of all Class 3 devices. So unlike the Class 2 devices, where you have to show that your device is as safe and as effective as something that's on the market, here, the PMA application must demonstrate safety and effectiveness, which is accomplished through design validation, manufacturing controls, performance testing, almost always performance of animal studies, at least for cardiovascular devices, and almost always clinical trials. But every device is different, and so these may vary. And finally, just for completeness, there's a humanitarian device exemption route to market. So these are devices that are for the treatment of a disease or condition that affects or manifests in itself in not more than 8,000 individuals per year in the US. So the HDE application is very similar in form and content to the PMA application. Remember, the PMA application for Class 3 devices, you have to show safety and effectiveness. Here, you have to demonstrate safety, but you only have to demonstrate probable benefit. Clinical investigations to generate effectiveness are not required. And the idea is that this is such a small population that the ability to do a full effectiveness study is really limited by the small population. And so you can forego that, but you do have to make an argument that there's probable benefit. There are restrictions on its use, most notably that it has to be used in facilities under IRB supervision. And as an example, this was the first one that was approved in 1996, which is a fetal bladder stent. So this is for a fetus where there's bladder outlet obstruction, so the fetus can't urinate, the pressure rises, and if left untreated, would damage the kidneys. So this is a stent that's placed in utero to relieve the bladder outlet obstruction, and then that allows the infant to grow normally, and then they can fix it once the child is delivered. So just a few comments on off-label use. We've talked about marketing and labels, but let's talk a moment about off-label use and development strategy. So the FDA is prohibited by law from regulating the practice of medicine. So their stated statement on this is that good medical practice and patient interest require that physicians be free to use commercially available drugs, devices, and biologics according to their best knowledge and judgment. Now, if a physician does this, it's really their responsibility to be well informed about the product and to make sure that they're basing its use on a good rationale and a sound medical evidence, and to keep a record of their use of that. So the use of a legally marketed device for an off-label indication, which happens every day, as part of the practice of medicine for an individual patient, it may require local IRB approval. That's not usual, but it can. But an approved investigational device exemption from the IDE is not required. And Alex will talk about IDEs in a moment. But I hasten to add that a clinical investigation, if you're doing a study, even if it's only one patient, that has to be conducted in accordance with the IDE regulations. So we've talked about devices that are legally marketed. So I want to also specifically note that an unapproved medical device can only be used on human subjects when the device is under clinical investigation and when it's being used by investigators participating in the clinical trial. The investigators must comply with all applicable requirements. So devices have to be legally marketed for use in humans. There are emergency exceptions. You can't simply make a new device and start using it in patients. And a legally marketed device that's modified may no longer be the legally marketed device. So if you buy a legally marketed device and you change it, add something, alter it, you probably aren't dealing with the legally marketed device anymore. It's now you've made it into an unapproved medical device. So this testing strategy, so this is very brief, single slide. There's, as I say, there's a complimentary talk on preclinical device development. And going through this slide, this represents possibly years and years of work. But I just wanted to, just for completeness, to review what goes into and the kinds of into developing your strategy and what the FDA will be thinking about. So you need to consider the intended use of the medical device, including the target population. Who are you going to be using the device on? To identify the appropriate testing, first of all, you need to identify potential safety and effectiveness issues through an analysis of the potential failure modes. You need to think through what can go wrong with my device. Can it break inside the patient? What if the pieces don't come apart? What if something gets stuck in there? What if whatever? Identify everything that can go wrong. And then during your development, you'll design your device to mitigate those failure modes and to address them. And so then you need to identify the design specifications. These are the characteristics of the device that are needed to maximize performance and to minimize the potential problems under the expected conditions of use. And from there, we would identify tests that will demonstrate if the design requirements are met. And these tests could be bench, animal, or clinical testing. And unlike in the development of a drug where you can't change a single atom without having a whole new drug, iterative design is the norm. And so during all of this process in developing your device, figure out what's wrong, fixing it, figure out what's wrong, fixing it, there's repeated cycles of design and iteration that mostly happens pre-market, but even once on the market or even in a clinical trial, issues may arise that require a design change. And because medical devices are multi-component products, many times those changes can be implemented without having to start from scratch as if it were an entirely new device. So let's talk about the investigational device exemption or IDE regulation. So say that if you're at the point where you need to do a clinical study or want to do a clinical study, then that's when the IDE regulations will come into play. I do want to note up front that this applies equally to manufacturers, like a large device company that's developing a product, as well as individuals, say a physician who comes up with a device idea and wants to test it in one of his or her patient populations. They apply to everyone, just like the IRB rules. And I also want to make the point that if I happen to say the investigational device exemption regulations apply, I'm not saying that you have to have FDA approval. I'm just saying there's rules you need to follow, just like there's IRB rules. So before I go into the decision tree, I want to make a few specific points. I did spend some time talking about class 1, class 2, class 3, and routes to market like 510K and PMA. I want to make sure you understand that the route to market is irrelevant to the applicability of the IDE regulations. Sometimes people say, well, it's a class 2 device, so therefore it's a study that I can do without FDA approval. And that's really not how it works. The IDE applications are applied and interpreted independent of the route to market and whether it's class 1, 2, or 3. That risk determination has nothing to do with the IDE regulations. What does matter in considering how to apply the regulations is whether a device is cleared or approved for marketing for any indication. And if it is, what is its indication for use, including the purpose and potentially the target population? And does it match your proposed use? So there's a decision tree that you need to go through, these regulatory questions that you need to go through in order to reach the correct decision about how to apply the IDE regulations to your study. First of all, is it a device? And we've talked about that. If it's not a device, then you're done with this question, with this decision tree. Presuming it's a device, you need to understand if it's already marketed and in commercial distribution, and if so, what are the indications for use? That will determine whether the study's exempt or not. So then you need to determine whether the IDE regulations apply to your study. Is the study exempt? And finally, if it's not exempt and is subject to the regulations, is the study, not the device, but the study as a whole, a significant risk or non-significant risk study? And it's important to do these in order. So sometimes people will start and they'll start trying to decide if it's a significant risk or non-significant risk study before they even figure out whether the IDE applications apply. Or having determined that it's an exempt study, I'll see IRB protocols where they decided it's exempt, but they continue to do a risk study. You go through the steps, and if it's exempt, then you're done. So I'm going to go through the different components of this, mainly these last components on how to go through the process. But first, the purpose of the IDE regulations is to encourage discovery and development of devices consistent with the requirements to protect public health and safety and with ethical standards of human studies. So when Congress enacted the medical device amendments, they recognized that in order to develop and discover new devices, those new devices had to be exempted from the requirements that normally apply to devices in commercial distribution. Remember, I said devices had to be legally marketed to be used in humans. But if you're studying a new device that's not legally marketed, well, you have to have a get out of jail free card on that. So what the investigational device exemption regulations do is they exempt the new device from all of these requirements. And these are, as from here on out, I have a number of links to different resources, but 21 CFR 812 is what covers the IDE regulations. So the conduct of a clinical investigation of a medical device requires approval under the IDE regulation. Now, that doesn't mean you have to have an approved IDE from the FDA. It may be approvable by the IRB, but it has to be approved in accordance with the regulations. And an investigation is defined as a clinical investigation or research which involves one or more subjects to determine the safety or effectiveness of a device. So sponsor investigator studies of unapproved devices, that would be a physician at a hospital who doesn't have a manufacturer involved, a sponsor investigator study of an unapproved device or a new intended use of an approved device, even if there's no marketing application planned, those studies are subject to the regulation. Now, I do want to mention that in the regulation of devices, it doesn't matter if the investigation will or will not be used to support a new indication for use, or if it will or will not be used to support a significant labeling change by the manufacturer. And I make this point only because those two things can be factors in the application of the investigational new drug regulations, but they have no bearing on the device, on the application of the investigational device exemption regulations. So I said one of the questions that you need to answer in going through this path is exemptions. So an investigation is exempt from the IDE regulations if the device is used or investigated in accordance with the indications in the approved labeling. So the FDA has judged the device to be safe and effective for its stated use in the label. So if you consider, suppose a cardiologist wants to do a comparative study of two stents, both of which are approved for the treatment of coronary artery stenosis or a blockage. So if they're both approved for the same thing, they're going to do a head-to-head comparison. The IRB rules would apply, and because it's a study, then the cardiologist would have to have IRB approval, but it would be exempt from the IDE regulation because they're being used in accordance with its labeling, so with no safety and effectiveness issue to be evaluated. In contrast, if an interventional neuroradiologist wants to use one of these same stents to study its use in the treatment of intracranial arterial stenosis or blockage in the setting of acute stroke, then the IDE regulations would apply because you're using a device which is legally marketed, but you're not using it in accordance with its labeling, and you're conducting a study of the safety and effectiveness. So here in the second example, the IDE regulations would apply, and we'd go on to the next question, which is significant risk versus non-significant risk studies. So a significant risk study is one which presents a potential for serious risk to the health, safety, or welfare of the subject. And the risk includes the risk of any ancillary procedures that are involved. So if you have to do a surgical procedure to use the device, then the risks of that surgical procedure may play a role in your determination of risk. And it's the risk of the study, not the device, that determines the regulatory control. So there's much more detail on this in the guidance sheet on significant risk and non-significant risk studies, including a checklist. There's 4 categories that it goes through that you need to consider in determining risk. So what's the protocol review approval process? So you've gone through your checklist and you've determined that it is a device study, and now you need to figure out what level of control is going to have. Is it going to be approved by the IRB or by the IRB and the FDA? So you as the sponsor or sponsor investigator in developing your protocol, you would provide a rationale for your initial assessment of risk. You would might argue that it's a non-significant risk device because, and define why that is. The IRB then will review your protocol, and they are responsible for determining whether it's a significant risk or a non-significant risk study. If the IRB determines that it is a non-significant risk study, then the IRB can then go forward and simply review it and approve it as they would any other study. Now, in this setting, the FDA delegates its authority to the IRBs to make this determination, and the IRB acts as their surrogate. If it's a non-significant risk study that the IRB approves, then it's subject to what are called the abbreviated IDE requirements, and those are all available on the FDA website. On the other hand, if the IRB determines that this is a significant risk study, then it will require both IRB approval and an approved or conditionally approved IDE application before the study can begin. So at the NIH, in our system, we would have to have FDA approval before seeking IRB approval, but at most institutions, you can pursue these simultaneously. So if you've developed a new heart valve and you want to study it, it's plainly going to be a significant risk study, so you don't have to wait for the IRB to tell you that it is. You can simply say this is a significant risk study, submit to both simultaneously, and the two of them can work it together if there's some discrepancy in what their requirements might be. If at any point the FDA determines that a study is significant risk or non-significant risk, the decision's binding, and that can go either way. I've had seen studies where it was sent to the FDA because the IRB thought it was a significant risk study, and the FDA said, no, it's a non-significant risk study. They send it back to the IRB, and it is a non-significant risk study because the FDA said it was. They are the overriding authority, overall authority. So if you have to submit an IDE to the FDA, There's a number of requirements. The highlights are there has to be a U.S. sponsor. The report of prior investigations is critical. In this section of the application, you develop the knowledge, present the knowledge base of what's known about the device, but it's essential that you include any information about adverse safety events that may have happened. You include your investigational plan, manufacturing information, and so on. And again, there's information on the web which deals with this on the IDE application. And this section also includes information on common errors in submitting an IDE application, what's omitted, what can cause them to get bounced back. And a great resource at CDRH is this page called Device Advice, Comprehensive Regulatory Assistant. It covers how to study and market your device. It has information on the IDE exemptions. And I worked at the FDA for more than 2 decades, and I will tell you that I use this a lot just in day-to-day work. So let's finally turn to interacting with the FDA if that ends up being something that you need to do. So there's what's called a pre-submission program, or abbreviated as a Q submission. And this is a way to get informal confidential feedback, and it's free, which is the best part. So it's generally about a 60-day review. I don't think that's a statutory requirement, but it can take a month or two. And this can happen in any number of ways, by letter, phone call, email. Perhaps the most productive way is a face-to-face meeting, which are usually about an hour. These are not required, but it gives you an opportunity to get informal FDA feedback to help guide device development. And this can be appropriate at any stage of the testing or protocol development. It might be preclinical when you're designing your, let's say you have bench testing about mechanical stability. It might be about your preclinical bench work. If you're developing a pivotal animal safety study, it might be before that. And if you're going into a clinical trial, it can include that as well as marketing. And again, there is guidance document on how to interact with the FDA through this Q submission program. It reviews what are appropriate questions to ask, how to interact with them, and how to make the most of your time when you are interacting with the FDA. I want to close with a comment on a couple of myths that I hear and objections, and just dismiss these. One is, I always hear people, don't talk to the FDA until you've submitted your application because, and any number of reasons, and I've never really quite understood that. I don't know if people are afraid that the FDA will tell them to do a test that they don't want to do, but far better to find out early rather than late. So the FDA, I just talked about the Q submissions, the FDA encourages dialogue early in device development to facilitate entry into clinical trials and marketing. As I said, better to find out sooner rather than later about concerns. It can save you time if you start something early, and it can certainly save you money if you find out that you don't have to do something that you were about to. And for investigational device exemptions, going through the Q submission process can improve the likelihood of approval on your first or second round of an IDE submission as compared to folks that just submit it without talking with the FDA. Again, it depends on the complexity and whether it's a truly novel device or something that you have a lot of experience with. But the Q subs can help you with that. And by all means, talk to the FDA early. I've heard people say the FDA would never approve of a study of whatever. And so I've been at meetings when where someone said the FDA would never approve a study of blank. And at the time when I was an FDA employee, I happened to know, it was strictly confidential, so I couldn't share it, but I happened to know that there were 2 approved studies that were studying exactly what the speaker said the FDA would never approve a study of. So you have to keep in mind that FDA submissions are usually something new. Everything they see is new. It's not marketed yet. We now have transcatheter heart valves that I mentioned earlier. If the people that were developing these some 15, 20 years ago had said, well, the FDA would never approve a catheter a valve that's being placed through a catheter in the groin. Well, if they had thought that, we would not have those devices now. My general advice on this is to simply let the science guide you. Figure out what you want to do, what you need to show, what you want your device to do, and let the science guide you, not your guess as to what the agency will or will not approve, either for marketing or for a clinical trial, and work with the agency to get your new technology developed and moving forward. And then finally, people say, I can't do this because the FDA's guidance document prohibits it. So I've mentioned several guidance documents already, and there are a lot, some of which talk about specific types of devices, some of which have suggested trial design, but guidance is simply guidance. It's not a regulation, it's not a rule. All that guidance is a presentation of FDA's view on regulatory requirements at the time the guidance was issued. So it's not binding on the FDA or a sponsor. If you follow the guidance to a T, the FDA doesn't have to approve your study. And if you prepare a protocol that hardly follows the guidance at all, the FDA could still approve it. It's not binding, it's just guidance. And it doesn't prescribe a fixed study design. Now, all of that said, if you're developing a study and there's relevant guidance that talks about that kind of product or that kind of disease or something, you would be well advised to review the guidance and follow it to the extent that you can. And where you can't, make sure that you use the guidance to be aware of the kinds of questions that the agency is going to ask. What kinds of things are they concerned about? What kinds of things do they see? Let it inform you, which is what the purpose of the guidance is, but you don't have to follow it. If you can, it's good. Let the science guide you. So just in closing, with when you're developing and testing your device, it's going to be an iterative design. You need to identify and address the failure modes, and then do testing that shows the design requirements are met, and that can include bench, animal, and clinical studies. For clinical studies, we talked about how to develop a clinical research protocol, and the questions in order: is it a device? Do the IDE regulations apply? Is it exempt? And if it's not exempt, is the study, not the device, significant risk or non-significant risk? And that will determine whether the IRB alone can approve your clinical study or whether both IRB and FDA approval are required. So in doing this, you need to use all the resources available to you. Local IRB, these usually have resources. Your institution may have protocol management teams. At the FDA, I've pointed out multiple times their website, and as I said, I use that almost on a daily basis. There's online training that they offer. There's education, different webinars. There are publications on regulatory matters, although you need to be cautious about interpreting that because not everyone who claims to be an expert is. And you can have direct FDA interactions as required. And beyond that, it may be useful to have someone who's had a number of clinical trials, especially device trials, under their belt to look at what you're proposing and to give you pointers and advice, or potentially read your protocol and identify any issues. So with that, I thank you for your time. And when we get to the live session, I look forward to answering any questions you may have.